Hello everyone, hope you're all doing fine. So today's topic is about protein and its structure. So the word protein comes from the Greek word proteos, which means first or primary. They were so named because by the J.T. Mulder, who thought that they should be ranked first and considered of primary importance among the chemical substances that control life. The proteins are the polymers of alpha amino acids. You all know about the amino acids. An amino acid is a bifunctional organic molecule that contains both a carboxyl group and an amino group in it. So there is different kinds of amino acids which make up the protein. There is hydroxy amino acids, thioamino acids, carboxy amino acids, and diamino acids, and aliphatic unsubstituted amino acids, heterocyclic amino acid, and so and so. There is different kinds of amino acid. So with the help of this amino acid, the proteins is made up of. So the proteins are an important class of biological macromolecules, which are the polymers of amino acids. Biochemists have distinguished the several levels of structural organization of protein. So when we discuss about the structures and shape of the protein, it is generally considered at 4 different levels: primary structure, secondary structure, tertiary, and quaternary, each level being more complex than the previous level. So here the structure is here. There is primary at the top, and then secondary, like alpha helix is shown here, alpha or beta pleated sheets. Then quaternary structure, tertiary structure. The quaternary structure, there is 4 subunits for it, as you can see that that is shown in red, orange, and blue and green color. So you can see the structures. So protein structure is a 3-dimensional arrangement of the atoms in an amino acid chain in the molecule. Proteins are polymers, as we said before. So specifically polypeptides formed from sequences of amino acids, the monomers of the polymer. Protein formed by amino acids undergoing condensation reaction, in which the amino acid lose one water molecule per reaction in order to attach to one another with a peptide bond. So when we say about the peptide, the product of condensation of alpha amino acid is called peptide. Okay, and the CONH linkage that unites successive amino acids or successive amino acid units in a peptide is called a peptide linkage or peptide bond. Okay, by condensation, like peptide can be according to the 4 or 5 or 6 or etc. amino acids condense. The proteins are respectively called tetrapeptide, pentapeptide, hexapeptide, and so on. So its condensation product is protein. To able to function or able to perform their biological function, proteins fold into one or more specific spatial conformation driven by a number of non-covalent interactions such as hydrogen bonding, ionic interaction, van der Waals forces, and hydrophobic packing. To understand the functions of the protein at molecular level, it is often necessary to determine their 3-dimensional structure. This is the topic of the scientific field of structural biology, which employs techniques such as X-ray crystallography, NMR spectroscopy, cryo-electron microscopy, and dual polarization interferometry to determine the structure of the protein. So moving on. About the primary structure. The primary structure is really a specific sequence in which various amino acids are arranged in a protein. Okay, that's a specific sequence in which various amino acids is arranged, and that is called primary structure. And this primary structure is so important that because it decides its biological activity. Hemoglobin, for example, it contains 574 amino acids, which is arranged in a very specific way. Okay, this primary structure is having the paramount importance to decide the biological activity of a protein. Then moving into the secondary structure. The conformation which the polypeptide chain in a protein assume as a result of intramolecular or intermolecular hydrogen bonding is called secondary structure. Okay, there is two common types of secondary structure is alpha helix structure and beta pleated sheet structure. Okay, there is another kind of structure like beta bends, alpha loops, ohm loops, like that. So I'm going to discuss about the main two common type of structure, that is alpha helix structure and beta pleated structure. So here there is a diagram of alpha helix. Okay, this is alpha helix. Okay, it was founded by 1951 by Linux Pauling, and this is the helical structure of that alpha helix. Sometimes the polypeptide chain in the protein coils up into a spiral structure. The turns of the coil are held together by the intramolecular hydrogen bonding, like you can see. Intramolecular hydrogen bonds between the C double bond O of one amino acid residue and NH of the fourth one in the same chain. So this type of structure is called alpha helix structure. The regularity in the hydrogen bonding accounts for the regularity of the turns of the coil. The axial pitch of alpha helix is 0.54 nanometer and it contains 3.6 amino acid residues. 3.6 amino acid residues. Alpha helical structure is found in many fibrous proteins such as keratin in hair, skin, nail and wool, and mucin in muscles. So when we start with here, that alpha helix has spiral structure, tightly packed coiled polypeptide backbone core, side chain extend outwards, stabilized by H bonding between carbonyl oxygen and amide oxygen, and amino acid per turn is 3.6. And the pitch is 5.4 angstrom. Alpha helical segments are found in many globular proteins like myoglobin, troponin, C, etc. And then the secondary structure. There are another kind of secondary structure. We already said that it is beta pleated sheet structure. So the difference between alpha helix and beta pleated structure is shown here. And the secondary structure is really the result of the hydrogen bonding happens in there. So moving on to the beta pleated structure or beta structure. Sometimes the long polypeptide chain lies side by side in a zigzag manner, each chain being held to its neighboring chain on either side by intermolecular hydrogen bonds to form a flat sheet. These sheets are then stacked one above another to form a three-dimensional structure. This type of structure is called beta structure or beta pleated structure. It is called as beta pleated sheet structure as it is resembling the pleats folds of the drapery. So the silk protein fibroin has the beta structure. It is also found by Linux Pauling in 1951. Okay, it can be two type, parallel chain, as you can see here. Parallel chain, the terminal NH2 pointing in the same direction, and the anti-parallel chain, the terminal NH2 groups will be pointing in opposite direction. Yeah, there is two types of beta pleated structure. And this is the diagram. And moving on to the tertiary structure of the protein. The tertiary structure of the protein is the three-dimensional structure which arises due to the further folding and superimposition of the different secondary structural units in a characteristic manner. This gives rise to the two major molecular shapes, namely fibrous and globular proteins. It is based on the molecular shape or solubility in the water. The protein can be classified broadly into fibrous and globular protein. Okay, fibrous will have the structure or shape similar to the fiber, and the globular protein will be in the globular shape. So fibrous protein, when we say about fibrous protein, it consists of long helical thread-like polypeptide chain which lie side by side or parallel and are held together by hydrogen bond and disulfide bond at many points to yield a fiber-like structure. So they are generally insoluble in water. Common examples are keratin in hair, skin, nail and wool, collagen in tendons, fibroin in silk, mucin in muscles, etc. And the globular protein are those in which the molecules are looped, twisted and folded together into compact units having ball-like structure. They are generally it is soluble The fibrous protein is insoluble in water. So globular protein are soluble in water. Almost all enzymes are globular protein. Other common examples are antibodies, many hormones like insulin, albumins, etc. So the tertiary structure of the protein, it is arises due to the further folding and superimposition of the secondary structural units in a characteristic manner, and that gave rise to the two molecular shapes, as we said before, fibrous and globular. And there is certain forces that stabilize this tertiary structure. The fourth type of forces which stabilize the tertiary structure is hydrogen bond, ionic or electrostatic bond, and disulfide bond or van der Waals bond. Okay, it is mentioned here. So hydrogen bond is seen between NH2 OH SH group in the side chain of one amino acid and the carboxyl group of other, and ionic or electrostatic bond formed between the charged side chain group NH3 plus or COO minus, and disulfide bond between the side chains of cysteine residues located in different parts of the polypeptide chain. As we know, cysteine have the sulfur in it. And van der Waals bonds or weak hydrophobic interaction between the hydrocarbon radicals of the amino acid. Okay, this is about the tertiary structure. This is the diagram of tertiary structure, which is having the sulfide bond. So the main forces which stabilize the tertiary structure is hydrogen bonds or ionic bonds or disulfide bond or van der Waals bonds. And the last one, quaternary structure. In the beginning of the class, I have showed you the structure of the quaternary here. Okay. The subunits is arranged here. Moving on to the quaternary structure. Some of the proteins are composed of two or more polypeptide chains, and that is referred to the subunits. Okay, this spatial arrangement of these subunits with respect to each other is called quaternary structure. For example, a hemoglobin is a protein which have the quaternary structure with 4 globular polypeptide tertiary structure, altogether giving again a globular conformation. Okay, the forces which stabilize this quaternary structure also include hydrogen bond, disulfide linkages, as well as van der Waals and electrostatic forces of attraction. So this is about the quaternary structure. So here you can see first you can see the primary structure, then you can see secondary structure, which is alpha helix and beta pleated structure there, then tertiary structure and quaternary structure, the arrangement of the subunits altogether. So this is the basic of the structure or structural classification of the protein. Hope you understand it. Thank you.